Welcome back to biology. My name is Mr. Kubuske. Today we're going to start covering Unit 8, which is all about genetics. So this is our honors biology class. When we talk about genetics, what we're really talking about is the work of a guy named Gregor Mendel. We're going to get to know him just a little bit and some of the work that he did. But I'm sure most of you, if you're watching this, if you're in this class, you've probably done something with genetics and something called Punnett squares before. Gregor Mendel did not create the Punnett square, but he did create the laws that govern how a Punnett square works, and that's what we're going to be talking about today. Okay, so Gregor Mendel and basic genetics. Okay, for example, let's say I've got two parents here. Okay, and we're going to talk about pea plants. Okay, and of these pea plants, I have a parent with purple flowers and a parent with white flowers. Okay, now take a second to make a prediction. What do you think their offspring would look like? Okay. Here's what happens. Okay, so what happened? I had a purple parent and a white parent, okay, and they made all purple offspring. So what happened to the white gene? Where did it go? Okay, well, let's take this a step further, okay? Let's take two of their offspring, and then we're going to cross them, okay? We're going to cross two of the offspring of the purple and the white parent. So we know that they got genes from the purple and the white parent. Even though they're both purple, what do you think their offspring will look like? Let's find out. Now you'll notice as they start to appear, okay, there are 5 of the 20, it looks like, that are, okay, white instead of purple, even though both parents were purple. So about a quarter of them ended up with the white genes. Now, how did that happen? Okay, so I had a purple parent and a white parent, and I got all purple flowers. Okay, I took 2 of those purple baby flowers, I crossed them, and I got 75% purple flowers and 25% white flowers. What the heck is going on? Where are all my genes going? How come things are disappearing and then reappearing? Okay, well, believe it or not, those are the questions that Gregor Mendel himself asked back in the 1800s when he was doing his work with pea plants. Okay. He was a botanist. He was studying these pea plants, okay, and what he stumbled upon, okay, were patterns and predictable patterns about these genes, these traits that would disappear and then reappear, okay, and from that he created rules and created laws that we follow today when we study genetics and determine inheritance, okay. But he was an Austrian monk. And don't think of him necessarily as being very religious. I mean, he was religious, don't get me wrong, but he was there to do work scientifically. Like, he was a botanist. He wanted to study plants. He because of his work with these science and these maths, okay, he was able to become what we call the father of modern genetics, okay? So he studied inheritance in pea plants. He noticed that traits would disappear and reappear, okay? And since he had dedicated his life to this monastery, to this work, okay, he was able to determine the patterns and the rules for these traits, okay? So let's talk about Mendel's rules. He's got 3 rules that you're going to need to know and then 2 laws. The first rule is every person has 2 copies of a gene. Where do they come from? Well, they come from the sperm and the egg, okay? You get one from your mom and one from your dad, okay? So here's your sperm and your egg, okay? For each trait, there are different alleles, or meaning variations of a gene, okay? For example, eye color, there's lots of different variations of a gene. For eye color, we call those each alleles, okay? For the plants earlier, the flower color, okay? We had 2 alleles. We had purple and we had white, okay? Now, how they combine, there could be different combinations of that purple and white, okay? But each individual letter then, okay, that we're going to use to represent them is called an allele. So we'll use capital letters and lowercase letters, and we'll determine how we figure that out here in a second, okay? Number 3, if there are different forms of a gene present, one of them will mask the other, meaning that one will be dominant to the other. It will cover it up. Even though the recessive one, the one that doesn't show up, is there, okay, since the dominant one is present, it covers it up. So even though in that first generation, and when we use the word generation, what we mean is parent to offspring, offspring to offspring, okay? Each time another set of offspring is produced, that's called a generation, okay? When Mendel did it, he called the original parents the F generation, and then each generation after that, he called it P1, P2, and P3. Okay? Just as a heads up if you see that anywhere that comes up. Okay? But the dominant trait, even though the recessive trait was there, the dominant trait covered up the recessive trait. Okay? And that's why we call them dominant and recessive. Now, we represent them with alleles, these letters, okay? And then the dominant one is always going to be the capital letter, okay? And the recessive one will always be the lowercase letter. But they'll be the same letter. Okay, now it's really easy when you're talking about things like eye color, like, okay, brown eyes is dominant to blue eyes, so it's big B and little b, okay? But we're really using the letter B because it goes with the dominant trait. Now, you can use any letter that you want just as long as you designate it ahead of time, okay? We could say that, since it's eye color, we're going to use an E, okay, and then big E will represent brown, and little e will represent blue. Okay, so you can use any letter, but the letter is going to be the same if it's dominant recessive. The only difference is one's capital, one's lowercase. Okay, so keep that in mind as we're going through here. Okay, so Mendel's two laws. His two laws are actually going to mirror what we just got done talking about in Unit 7. Okay, the law of segregation. What that means is that for each trait, okay, we've got our chromosome here, our homologous chromosomes. Okay, one from mom and one from dad. Okay, and what happens to them during meiosis, we should know by now, is that they're going to separate. Okay, one will end up in this gamete, and one will end up in this other gamete. So my big A and my little a will be in two different gametes. That's the law of segregation. They separate. Say segregate, separate. It means kind of the same thing. Now, how they separate is governed by the next law, the law of independent assortment. Law of independent assortment says that alleles of different genes separate independently of one another. Okay, so in our diagram here, we've got our A's and our B's on different chromosomes. Does it matter if it's capital A and capital B? Do they have to go together? No, they can. It could happen, okay, but that's not the way that it's necessarily always going to be. I could put a capital A in this one and a lowercase b, okay? And on the other side, I could put a little a and a big B. Or I could do a little a and a little b, okay? There's no rhyme or reason to it. It's completely random, okay? So that's what we mean by independent assortment, okay? Just because I use a capital for one doesn't mean it has to be a capital for the other. Independent assortment. They assort independently, okay? So, using Mendel's rules, we can make predictions about what will happen before it happens, but we use something called a Punnett square in order to do that. Now, again, Punnett squares were not created by Gregor Mendel. The laws that govern Punnett squares were. Okay? Now, all we have to do to make a Punnett square, and all it's going to do is give us predictions. Okay? It's not a guarantee. It's not saying that if you have 4 kids, they will all end up with one of each of these boxes. Okay? It's just a predictor. It's for statistics and things like that. Okay, a Punnett square is just a box with a cross in it. And I know it might be hard to see with my red. I'll switch over to black here from now on. Okay, but that's how we create a Punnett square. Now, how do we actually use it? Okay, let's take one of the other traits that Mendel studied. Okay, besides his pea plants and colors, okay, he studied the actual peas themselves. Okay, so there was the round ones which showed up more often, which told him that they were dominant, and then there were wrinkled ones which would show up and then disappear and show up and disappear. Okay, so we're going to say that the round is dominant. So we've got a round parent, big R, and a wrinkled parent, little r. Okay, those are the alleles we're going to use. Okay, so what I'm going to do now with these two parents is I'm going to put them in specific places on this Punnett square in order to make predictions about their offspring. But the way I do it is by following both the law of segregation, which means the R's will separate from mom and dad, and that they will independently assort. So I could put either one with either one. I just realized I had that in my back pocket. Okay. So for the first R, for the first parent, big R, okay, I'm going to take his first letter, his big R, and I'm going to put it above the first column. And then his second letter, okay, I'm going to put above the second column. And then after I do that, I can bring each of those letters down, and they will be represented in all the boxes below them, straight down. I do the same thing with the second parent, okay. I'm going to bring the first R, and it'll go into the first row. And then I bring the second R, and I put it into the second row. Now, it just so happens that the letters are the same, so it really wouldn't necessarily matter which one went where. Okay? But then I bring the information straight across. Okay? So no matter what will the seeds of these offspring look like? They will all be round. How do I know that? Because each one of them has a capital R in their box. Okay? So no matter what, it's a 100% chance that these parents will have round seeded offspring. But what if I take 2 of those offspring that I know their letters now is big R, little r? What if I took 2 of those and I crossed those? So big R, little r for both parents, okay? Well, first thing I gotta do is I gotta make my cross. And now I'm gonna place my letters, okay, my alleles. Again, first one above the first column, second one above the second column. Okay, and I can bring that information straight down. Big R, little r. The same thing with the second parent, okay, but instead of doing the columns, I'm doing the rows now. Big R, little r. Now, if you do this correctly, you'll see I have the little r in front of the big R. If you do it correctly, the capital letter should go first, okay, but it's not the end of the world because as long as there's a capital letter in that box, that means it's going to have the dominant trait, okay? So if I look at my 4 squares, what are the chances that my offspring will be round seeded? Well, all I have to look for is that capital R. Okay, there's a capital R in this box, so there's 1 out of 4. There's a capital R in this box, that's 2 out of 4. There's a capital R in this box, that's 3 out of 4. There's no capital R in that last box, so 3 out of 4 means I have a 75% chance. Just think of it like quarters. I have 3 quarters, okay? That's 75 cents. Okay, 75% works the same way. Okay, so what's the chances that my offspring will be wrinkled seed? The only way they can be wrinkled is if they have 2 lowercase r's, okay, 2 little r's, and there's only 1 out of 4 boxes for that, so that would be 25%. You can always check your work because the percentages should add up to 100% if you do it correctly. Okay? Take a second while I erase this. See if you can do the brown eye, blue eye example. Okay. So of these parents, what's the chances that their kids will have blue eyes? 0%. Why is it impossible? Well, because each one of the kids is going to get a capital B from the first parent, which means every one of their letters will have a capital letter, meaning it's dominant. Okay, good. Now, there's some terms that are associated with these Punnett squares and with the study of genetics that you're going to need to know that are going to help you answer problems, especially long, like story type problems, where they kind of give you, they tell you the family history and they tell you a little bit about the parents and what they look like. Okay, but you might not necessarily know the exact genes of them, but you can determine them just by using some of these terms. Okay, the first 2 you need to know are genotype and phenotype. Okay, genotype is the actual genes; it's the letters. Okay, so big B, little b, that's a genotype. Okay, they represent the genes; they're the alleles. Okay, they represent the gene that you get from your parents. So genotype is the actual genes themselves. Okay, what it looks like. Okay, or I guess it'd be brown eyes if we're going to use that example. Okay, something else. Okay, what it actually looks like is called a phenotype. Okay? Phenotype for physical features. Phenotype, physical features. So genotypes and phenotypes. I can determine those with Punnett squares. Okay, so is this a genotype or a phenotype? Obviously, genotype, phenotype, genotype, genotype, phenotype. There you go. Okay. The next 2 that you need to know are homozygous and heterozygous. Okay. Homozygous has the prefix homo, which means same. So homozygous means that the genotype, the letters are exactly the same. They're both capital or they're both lowercase for these examples. Okay. So how many homozygous plants do we have in this example here for seed type? We have 2. The capital R, big R, and the little r. Those are both homozygous. Now, it's 2 different phenotypes, but the genotypes are both homozygous. They just happen to be different letters, okay? Heterozygous, the prefix hetero means different, okay? So the letters are going to be different. One will be capital, one will be lowercase. So this one, for example, is heterozygous because it's big B, little b, okay? Now, homozygous is sometimes referred to as purebred, and heterozygous is sometimes referred to as hybrid, meaning a mixture, okay? So this is homozygous, homozygous, heterozygous, heterozygous, homozygous. Okay. So we'll do one more example here together. Okay, and then these are going to be kind of the ways that you're going to be asked these questions. You're going to be given a little bit of a family history, and you're going to have to determine, okay, the family and the information about the family from that information. Okay, so I've got dad that's heterozygous. Okay, well, what does that tell me about dad? Okay, if we have brown eyes is dominant to blue, that means dad has to be a big B and a little b. And I don't know if you can see that purple or not. Okay. Mom is blue eyes, so I don't even need to know that she's homozygous. I can already figure that out because only way blue eyes shows up, since it's recessive, if it's a little b, little b. Okay. Now, what are the possible phenotypes of the children? Okay, I bring dad's letters down and mom's letters across. There's only 2 possible genotypes, but how many phenotypes is that? Well, let's figure it out. Okay. These 2 have a capital B. The only thing that they can be is brown eyes, so it's 50% chance for brown eyes. And the only thing these 2 could be, since they have 2 lowercase b's or little b's, is blue eyes. So there's a 50% chance the kids will have blue eyes. Okay? So the 2 possible phenotypes are brown eyes and blue eyes. Okay? But if they were both blue-eyed, the only possibility would be kids with blue eyes because no matter what, they get little b's. Okay? Now, believe it or not, this unit is not heavy on vocabulary. This is actually probably pretty much it. There might be a word or 2 that go along with some of the more advanced genetics concepts that we're going to cover later on, but this is pretty much it. You need to know Mendel, know allele. Remember, those are the letters. Those are different forms of the same gene. Dominant covers recessive. Segregation means separation. Independent assortment. Big A can go with little b. Genotype is the genes, phenotype, physical features. Heterozygous different, homozygous same, and then a Punnett square is what we use to make predictions, okay, about possible inheritance. Okay, remember it's just a prediction; it is not a guarantee.